The Babraham Institute's research has devised a mechanism to time-leap human skin cells by 30 years, effectively turning back the clock for cells without losing their specific function. Researchers from the Institute's Epigenetics Research Department have been able to partially repair the function of aged cells while also regenerating molecular measurements of biological age. The study was published last week in the journal eLife, and while it is still in its early stages of development, it has the potential to transform regenerative medicine. Welcome to today's episode of Longevity News. In this episode, I will show you one of the new abilities the Yamanaka Factors have been shown to have and how you're soon going to be able to profit from it. Our cell's capacity to operate reduces as we age, and the genome accumulates aging markers. Regenerative biology seeks to repair or replace cells, even those that have died. Our capacity to generate induced stem cells is one of the most essential tools in regenerative biology. The procedure consists of numerous phases, each of which removes some of the markers that differentiate cells. In principle, these stem cells have the capacity to become any cell type, but scientists have yet to successfully reproduce the circumstances that allow stem cells to re-differentiate into all cell kinds. This is done with the help of the Nobel Prize-winning Yamanaka Factors. The Yamanaka factors are a collection of protein transcription factors that play an important role in the development of induced pluripotent stem cells, cells that may become any cell in the body, often known as iPSCs. They regulate how DNA is duplicated in order for it to be translated into other proteins. They are also known as OSKM genes. Professor Shinya Yamanaka created iPS cells from human adult fibroblasts in 2006, and after starting with 24 transcription factors known to be critical in the early embryo, he and his research team narrowed it down to 4 major transcription factors. The factors are extensively expressed in embryonic stem cells and can produce pluripotency in human somatic cells when overexpressed. These factors control the developmental signaling network that is required for embryonic stem cell pluripotency. Yamanaka factors are frequently employed to turn adult cells into induced pluripotent stem cells. These have become key tools in longevity research, but they are a double-edged sword, since overexposure can result in the erasure of a cell's identity. Yamanaka was awarded the Nobel Prize in Physiology or Medicine in 2012, together with John Gordon, for discovering that adult cells may be transformed to stem cells, and in 2013 he was given the $3 million Breakthrough Prize in Life Sciences for his work. Using the Yamanaka factors, we may be able to turn back the clock, shifting from aging research to epigenetic rejuvenation research. The new procedure, which is based on a Nobel Prize-winning technology used by scientists to create stem cells, addresses the challenge of completely deleting cell identity by stopping reprogramming partway through the process. This enabled researchers to strike the perfect balance between reprogramming cells, making them biologically younger, and restoring their specialized cell function. Shinya Yamanaka was the first scientist in 2007 to convert normal cells with a specified purpose into stem cells with the capacity to grow into any cell type. The entire process of stem cell reprogramming takes around 50 days and involves 4 critical molecules known as the Yamanaka factors. The new technique, known as maturation phase transient reprogramming, exposes cells to Yamanaka factors for just 13 days.
And what we found, and this was a remarkable day, my student texted me the result. You could see that the nerves were growing back towards the brain. In fact, if we leave them for 16 weeks, many of those nerves actually make it all the way back to where they meet the brain again, the chiasm, which is a first. This hasn't been done before. There's very little you can do for damaged nerves. But then we looked at the age of those cells, and 2 interesting things came out. One was that the damage accelerated aging according to the Horvath clock, which we didn't expect. I mean, who knew that damage would accelerate aging, but it does. And then the reprogramming factors delayed and reversed that age so that they became young and could grow back again.
Age-related alterations have been erased at this stage, and the cells have momentarily lost their individuality. The partially reprogrammed cells were allowed to develop normally for a period of time to see if their unique skin cell function restored. Genome analysis revealed that the cells had recovered skin cell markers, fibroblasts, which was corroborated by monitoring collagen synthesis in the reprogrammed cells. The researchers sought for alterations in the signs of aging to demonstrate that the cells had been revived. Our understanding of aging on a molecular level has progressed over the last decade, giving rise to techniques that allow researchers to measure age-related biological changes in human cells, explained Dr. Diljit Gill, a postdoc in Wolf Rake's lab at the Institute who conducted the work as a PhD student. We were able to use this in our experiment to measure the level of reprogramming achieved by our novel approach. The researchers looked at a variety of cellular age measurements. The first is the epigenetic clock, which uses chemical markers found throughout the genome to determine age. The second component is the transcriptome, which contains all of the gene readouts produced by the cell. When compared to reference datasets, the reprogrammed cells matched the profile of cells that were 30 years younger by these two metrics. The prospective uses of this technology rely on the cells not just seeming younger, but also behaving as youthful cells. Fibroblasts create collagen, a substance present in bones, skin, tendons, and ligaments that aids in tissue structure and wound healing. When compared to control cells that did not go through the reprogramming procedure, the rejuvenated fibroblasts generated more collagen proteins. Fibroblasts also migrate to places that require healing. The partly regenerated cells were examined by making an artificial incision in a layer of cells in a dish. They discovered that treated fibroblasts migrated faster into the gap than older cells. This is a positive hint that one day, this study might be utilized to make cells that repair wounds more effectively. This discovery might lead to new therapy options in the future. The researchers discovered that their technique had an influence on additional genes connected to age-related disorders and symptoms. The APBA2 gene, which has been linked to Alzheimer's disease, and the MAF gene, which has been linked to the formation of cataracts, both exhibited alterations toward younger levels of transcription. The mechanism behind effective transitory reprogramming is yet unknown, and this is the next piece of the jigsaw to be solved. The researchers think that crucial portions of the genome involved in cell identity formation may be spared from the reprogramming process. Our findings reflect a significant advance in our knowledge of cell reprogramming, Diljit said. We have demonstrated that cells can be renewed without losing function, and that rejuvenation attempts to restore some function to elderly cells. The fact that we also detected a reversal of age signs in genes connected with illnesses bodes well for the future of this research. This finding has extremely intriguing implications, said Professor Wolf Rake, a group leader in the epigenetics research program who just transferred to oversee the Altos Labs Cambridge Institute. We may eventually be able to identify genes that regenerate without reprogramming and particularly target those to combat the consequences of aging. This technique offers great potential for crucial discoveries that might pave the way for a fantastic therapeutic horizon. Van Vliet and her smart colleagues previously shown that she could sort MSCs using a unique microfluidic technique that can identify the 20% that drive blood cell proliferation. She and her students, on the other hand, intended to improve on that by figuring out how to stimulate a whole population of MSCs to create the essential components. To do so, they had to first determine which criteria were most significant. They discovered that, whereas numerous variables influence blood cell development, the production of a protein known as osteopontin was most strongly associated with improved survival rates in mice treated with MSCs. The researchers next looked into mechanopriming the cells to get them to create more of the required components. Van Vliet and colleagues have demonstrated over the last decade that altering the mechanical qualities of surfaces on which stem cells are cultured may impact their differentiation into mature cell types. However, in this work, she demonstrated for the first time that mechanical qualities can influence the substances secreted by stem cells prior to committing to a certain tissue cell lineage. Stem cells extracted from the body are often cultured on a flat sheet of glass or rigid plastic. The MIT researchers decided to try growing the cells on a polymer called PDMS and varying its mechanical characteristics to observe how it affected the cells. They created materials with varying stiffness and viscosity, which is a measure of how rapidly a material expands when stressed. MSCs grown on materials with mechanical qualities most comparable to bone marrow produce the highest amount of the components required to trigger HPSPCs to develop into mature blood cells, according to the researchers. The researchers next put their freshly developed MSCs to the test by implanting them into animals with irradiated bone marrow. Despite the fact that no HSPCs were implanted, this treatment swiftly repopulated the animal's blood cells and helped them recover faster than mice treated with MSCs produced on typical glass surfaces. They also recovered quicker than mice treated with the microfluidic sorting device selected factor producing MSCs. The researchers also want to see if mechanopriming may cause MSCs to create other factors that drive the formation of new cell types that could be used to treat other disorders. All this shows us that using Yamanaka factors to reverse aging is clearly possible, but there's still a long road ahead of us to do so safely, without actually causing cancers or other problems. So, what is your opinion on using Yamanaka factors to reverse the aging process? Do you believe the hype, or is this just going to be forgotten in a few years? Please tell us your opinion in the comments section below. I would love to hear what you have to say about it. Thank you for watching Longevity News. We consistently report on the newest biotechnologies and supplements around longevity on society's path to curing aging. Please support our cause by liking this video and subscribing to not miss out on any new videos.